It is such a privilege and a pleasure to be here to introduce a true icon in our field. I've been asked to make 2 announcements before that. The first is that the code for continuing medical education credits is 66836. I'll repeat that, 66836. Also, for those of you watching remotely, you can submit questions to the speaker anytime during the lecture. Just click on the button that says Send Live Feedback, which is below your videocast window. Type in the message and we'll relay it to the speaker via Chris in the front row here at the end of the lecture. There is so much that I could say about today's speaker, but because I know that she has so much to tell us about her exceptional work, I'm going to keep it just to the basics. Dr. Akil is the Gardner-Quarton Distinguished University Professor of Neuroscience and Psychiatry and a Senior Research Professor at the Michigan Neuroscience Institute at the University of Michigan. She is internationally renowned for her foundational research in understanding the neurobiology of emotions, particularly pain, stress, depression, and addiction. Her pioneering work on endorphins and their role in stress and pain led to absolutely groundbreaking discoveries. She continues to explore genetic and neural mechanisms driving mood and emotional disorders, bridging neuroscience and behavioral studies, and bringing critical and clinically relevant information into our knowledge of how the brain responds to challenge, which is so very relevant today. I personally can tell you, and I really want to emphasize that she is a tireless and dedicated mentor and advocate for our field and for those in our field. She is the matriarch, I hope you don't mind me referring to you that way, of a large lineage and family tree of students who in their own rights have become incredibly successful and influential. She's also at the other end, a less local end, promoted global awareness of mental health and brain disorders through her work at national and international organizations. Dr. Akil has received many prestigious awards and honors, too many for me to mention here, but one that I want to emphasize and highlight is our nation's highest science award, the National Medal of Science, which she received from the President in 2023. She's past president of the Society for Neuroscience, an elected member of the National Academy of Sciences and the National Academy of Medicine, and a fellow of the American Academy of Arts and Sciences. Friends and colleagues, Dr. Huda Akil.
Good afternoon, and thank you so much, Karen, for this wonderful introduction. I am really not only honored, but really thrilled and grateful to be here, specifically to be giving the WALS lecture at the NIH, which has played a huge role in my life. NIH in general and these 2 institutes, NIDA and NIMH, have supported my entire career. Back 50 years ago, in the middle 70s, my husband was starting a residency at the university at Stanford, Stan Watson, and he was excited about going there, so I decided to find a postdoctoral fellowship there. And Dr. Jack Barkus was very generous and took me in his lab, but his lab was a monoamine lab. And I was really interested in pain and the beginning of endorphin research. And I really wanted to work on that, and he was very generous, but he didn't have the grant for it. So I wrote a proposal to, back then, a new institute called the National Institute on Drug Abuse. And to my shock, they funded me. They funded my postdoc. I think I might be one of their first postdocs. And they allowed me to pursue this line of research that has defined my entire career and actually brought Jack's lab into that field of neuropeptides as well. So thank you, Naida. And then fast forward a few years, I got very interested in the stress system, the HPA axis, pro-opium melanocortin, CRF, and we were doing both fundamental research but also interested in translational research. And the National Institute of Mental Health not only funded some fundamental research, but allowed me to be the PI along with the right collaborators on human research, which again was a leap of faith that I am very grateful for, that allowed me to, while being focused on fundamental research, always keeping an eye on translation. So these two institutes and this little vignette is to just tell you the kind of influence and transformative changes that NIH has on the lives of all of us, on myself, on my students, on my colleagues, even my children who are now scientists. But more importantly, how much NIH has transformed really global knowledge and global health. Both the intramural program that does groundbreaking research, I've been hearing some of it today, it's really amazing, both from senior and junior people, and the amazing people in the extramural program who hold our hands, guide us, help fund us, and navigate all kinds of challenges, financial and otherwise, through thick and thin. Thank you all. You really are heroes, and what you do really changes life, in whether people know it or not, by bringing knowledge globally and saving lives globally. So honored to be receiving this award, this WALS opportunity. Thank you all. So I want to move on to talk a little bit about whom the real people are who are doing the science. And all the work that I do is in collaboration with my husband, that same said husband from 50 years ago, and collaborator, Dr. Stanley Watson. And our team, we call them the WAKILs, for Watson and Akil. Here's our names. I hope I will remember to mention them as I go forward, but look for them at the bottom of every slide. Some past WAKILs that have contributed to the talk today, and then some amazing collaborators, some of them at the University of Michigan, some of them elsewhere. A. Palmer in particular is highlighted because I'm going to talk about a project that involves him. He's at UC San Diego. I'm also part of a remarkable, I think, group of people who make up the Pritzker Neuropsychiatric Research Consortium, Stanford, Irvine, Cornell, Hudson Alpha Institute, and University of Michigan, where we're interested in looking at humans and the causes of psychiatric illnesses, both through postmortem and genetic and genomic analyses. Long-standing, amazing collaboration, and an equally rewarding and exciting collaboration through the Hope for Depression Research Foundation. They have funded some of the research that I will be talking about, and you can see also the remarkable colleagues who are all interested both in basic science and translational research in the field of depression. So, when you get to be my stage in life, you try to think big thoughts. And the thing that is on my mind the most is what I call the second pandemic, which is the current mental health crisis. I think it's been brewing for a while, certainly since the start of this century. It's been accelerating, but the pandemic definitely worked as a further accelerant. It triggered huge increases in depression and anxiety immediately. It interacted with the substance use disorders, including the opioid crisis. It triggered its own disorders, like long COVID being one of them, and it affected people, especially in disadvantaged populations, tremendously. And I believe we are still living in the shadow of this huge event that happened to humankind. And there are even very simple measures of that. So this is a simple Gallup poll in 2019. It asked how many people had a history of depression or were currently depressed. And then same question in 2023. In this 4-year gap, there was like almost a 50% increase in incidence of depression. And what concerns me the most is how much it's affecting young people. So you see, this is over a course of 20 years. Females' mental health status has really strongly deteriorated. Young women below the age of 29. In men, it's a bit slower, but it's actually getting a hit. And that's compared to old people who have been holding pretty steady. So there's something really to think about, that the mental health crisis is unevenly distributed and young people are really hurting. And this is, of course, deadly. So the rates of suicide have been going up in an alarming way. And in 2023, 1 death every 11 minutes. In parallel, there has been this, as we all know, this huge substance use disorders crisis. In 2023, 1 of 6 Americans, or 49 million people, had suffered from some kind of substance use disorder, a lot of alcoholism. And important to me and in my thinking is the combined overlap between substance use disorders and mental health psychiatric disorders. And here again, really deadly. So you could see through the course of 10 years between 2015 and 2025, the rates of death by overdose climbed alarmingly from 48,000 to a peak of 112,000, just breathtaking increase. They've begun to drop, but not nearly enough. So these are 2 huge escalating crises that are overwhelming this country and are actually global. And I think about, are they related? And importantly, as a scientist, how can science begin to help? So as a neuroscientist, I always put the brain in the middle. That's the center of life, of the universe. But I know that we know a lot about it at the circuit, cellular, molecular level, DNA level. So obviously we think bottom up. But it's also equally important to think top down and remember how much the environment matters, the general global environment, the family, the person, and how behavior in turn changes everything all the way down to the single gene in terms of epigenetics. And so when I think about these 2 disorders, in my mind, stress biology is key to seeing the link between them. Writ large, stress biology, writ large, has a clear role in both affective and addictive disorders. There is strong evidence of stress as a trigger, but also as a mediator of a lot of the consequences of these disorders on the brain. And again, in my thinking, the stress system represents a good framework for science to begin to grapple with these crises from a scientific standpoint, in a way, the same way that cell biology is a good anchor point, say, for cancer research, to give it as kind of a fundamental knowledge. And this is a system, even though the term stress seems kind of very vague and mushy, we know there's beautiful biology associated with this system. We know a lot about it, both in the brain and obviously in the pituitary, the adrenal, how it gets triggered, how CRF gets released, ACTH gets released, glucocorticoids get synthesized, how they have impact all over the body, and how they go back and affect negative feedback. We know the molecular player, and one of my most favorite and one of the first molecules I studied is pro-opiomelanocortin, which is like a signal in itself because it encodes both a stress hormone and beta endorphin. It has stress and a basis of drug abuse in one molecular stretch, telling us how these things are all related. So we know a lot about this system, but we really do not fully understand how it plays out in the brain and how it participates and triggers these dysregulations in these motivated behaviors and psychiatric disorders. So today, I wanted to kind of bring up 2 concepts that I feel that I'm thinking about, at least. One is kind of an expression that I made up, but it's not a very hard expression. I call it stress fitness, to be parallel in a way to physical fitness, something that should exist and that we should think about and strive for. And the other is the concept of resilience, not just as lack of sensitivity to stress. So I'd like to touch on those a bit today. So stress fitness in its simplest way is a healthy neuroendocrine stress response. But it goes a lot further, and I hope as we talk it will become evident. But at the simplest is a nice stress ACTH response, followed by court, followed by rapid negative feedback that shuts it off. So it needs to be sharp, it needs to turn off. And you can think of that kind of biology as a signal, as an alert, but a reminder that stress is not just like a fire. It's something that we do all the time. We're constantly monitoring our environment. It's an ongoing function where if things deviate from expectation, we notice them, we cope with them, we learn from them, we adapt so we can predict better, cope better in the future. So it's absolutely essential for survival and adaptation. But the system can go awry. It can either get facilitated and over-respond or last a long time and not shut off, or underperform. So your alert can become an alarm that doesn't shut off and become really annoying, or an alarm that doesn't ring loud enough, strong enough, long enough, so it doesn't prepare you correctly. And psychiatric disorders in particular, depression, anxiety, PTSD, have some of these hallmarks. Like PTSD is associated with under-responsiveness. Again, you all know this, but a reminder that not all stress is bad. There is so-called good stress. Low levels of stress are, in fact, essential. They're on the upswing of this U-shaped function. They increase synaptic functions, increase neuroplasticity, learning. They're important for engaging the immune system. But too much stress flips it over, and you start getting suppressed functions, reduced learning, and narrowing of attention, and possible even neural damage. And we all know the idea of allostatic load, that when it's chronic and never rests, the system never rests, then it becomes really deleterious even at a structural level in the brain. That's something Bruce McEwen in particular emphasized. And so an important idea then is, if we believe that there is a generally healthy way for our stress system to function, to be fit, can we begin to understand that and sort of hone it to who the individual is? Because not everybody is going to achieve stress fitness in the same way to prepare them to cope with psychiatric or addictive disorders. The problem is that stress is not an absolute thing. So one of the grandfathers of the field, Hans Selye, said, stress is not what happens to you, but how you react to it. And there are a lot of individual differences in how we perceive stress. And while that is really evident to anybody who does clinical research, in basic science research, we don't necessarily love thinking about noise or variability. And I think that for us to understand and wrap our mind around how we promote better ability to cope with stress, we need to confront that. So, as you know, the stressors can vary. The path to stress can be threat and uncertainty. We all know that if you don't know what's going to happen or it's threatening, it can be very stressful. Trauma, social context, but physiological stressors matter a lot. But all of this is impacting individuals differently, depending on their genetics, their development, their experience. And all of this translates into temperamental tendencies that I want to talk about a little bit. And it's that interaction that leads to either a strong adaptive positive reaction behaviorally and physiologically, or a negative one that involves negative coping. And both results in remodeling the brain in neuroplasticity, but it can be either good neuroplasticity, better learning, better adaptation, or bad neuroplasticity that sets the stage for stress-related disorders. So I want to take 1 minute and talk a little and mention experience-induced neuroplasticity, just to say that we don't think about experiences, specific experiences, at least again in neuroscience, how much they have opened the door for rewriting the programs in the brain. We've become really interested in this in a couple of domains. One, in the context of sensitization to opiates, that's the work of Wei Kumar et al., and the other is in the context of chronic social stress, the Eric Nestler model, social defeat stress, that's Murrah et al. Work. And we're using this approach called FOSS trap, where you can label a cell that's activated by an experience and letting it continue, like letting it marks it, and the animal can live on. You don't have to sacrifice the animal. And then you can let the experience continue with repeated exposures or administration, and then look at the endpoint with regular FOSS. So in this image, FOSS trap is a labeled protein in red, DT tomato. Green is regular FOSS. And when you see yellow, you say, in that brain, that same neuron got activated the first time and the last time. And when you look at that, you can say, what are overall the brain, all the neural networks that got activated the first time you encounter something, the last time you encounter something? It's painstaking, and as I said, I don't have time to get into it. But I want to tell you something overall that I learned. First of all, that a lot of experiences that you think should be specific show up brain-wide if you look at them brain-wide. But also, there is this initial response that biases the system in certain directions, and then repeated experience changes some systems, some nodes, and leaves other nodes alone. But there is this whole brain reorganization that comes from repeated experience that I feel we need to get under and understand and figure out where we can interfere before it solidifies into a permanent change. But I want to spend a good chunk of time talking more about the more initial part, which I'm calling temperament or sensitivity or resilience to stress. And I'm going to start with this question, which is, what is your idea of fun? Is it to sit at home knitting or to go skydiving? This seems like a silly question, but it captures a continuum that relates to different types of emotional reactivity that can be either more inhibited or more active, proactive. And when people, when they are at the extremes, they are prone to different types of psychiatric disorders called internalizing disorders versus externalizing disorders. Internalizing being depression, anxiety, and the like, and externalizing being conduct disorder, ADHD, some kinds of substance use disorders, and the like. So a lot of genetics is starting to think more about that kind of dimension versus gene-specific for like depression versus anxiety. Piazza, back in 1989, took regular rats, Sprague Dolly rats, put them in a novel environment, and asked how much do they explore. And some animals, he called the low responders, those knitters in red here, are scared and just hug the wall. I mean, this experiment we replicated. And then there's these highly exploratory animals that are all over the place, very excited about this environment, a new environment. And he showed that that is predictive of self-administration of psychostimulants, that low responders don't like to take drugs and high responders love to self-administer drugs of abuse. So early on, Mohammed Kabaj in my lab, we did this study because I wondered if it was so cut and dried and how it interacted with stress. So here is outbred animals split in half, high and low responders based on this behavioral test in a new environment. And in green for go, red is stop, like traffic lights. The high responders took cocaine very readily compared to the low responders. But if you expose these animals to psychosocial stress, social defeat stress, ahead of the self-administration, what you see is there is no longer a difference. And that's not because the high responders change. The ones that love that drug still love the drug. It's the low responder animals that usually don't love the drug, but now, after they've been stressed, they start taking drugs. So that led us very early on to this idea that there is these two paths to drug-taking behavior, a sensation-seeking, novelty-seeking approach, and a social stress-triggered drug-seeking. And we thought this cannot be doing exactly the same thing to the brain or interacting with the drugs the same way. But we wanted to understand this biology, so we set out to do selective genetic breeding on high and low responders to ask if that tendency was genetic or not and what we could learn from it. So John Stead in my lab started with very outbred as we could make it, tried to avoid inbreeding, so we wanted the heterogeneity. And we bred high to high, males and females, low to low, and you can see it diverged very quickly. These are different generations to the point that the low responders were hardly budging. So we changed the breeding plan a little bit, but basically highly heritable. If your parents are high responders, you're going to be high, and if they're both low responders, you're going to be low. So what comes along with that tendency that we looked at in this very simple behavior? So first of all, in a simple test of anxiety, the elevated plus maze, you can see that the low responders never venture out in the open arm. Even if they spend as much time exploring, running around the closed arm, in the open arm, they barely stick their nose out. But if you give them benzodiazepine, they go back out. So they're acting anxious. Similarly, this is from Chidam Aiden, where she did chronic unpredictable stress, and then looked at social interaction and found that in the inhibited knitters, if you will, low responders, there was a significant drop in social interaction after stress that you do not see in the high responders. And again, antidepressants reverse that. So you can see this kind of precision approach to antidepressants in these animals. Different stressors had different impact on them. And there is a whole bunch of other tests that we have done over the years, including fear conditioning, fear extinction, multiple publications, saying that we have kind of an inhibited phenotype in these animals. On the other side is the drug abuse side. If you just simply take animals and give them cocaine, you are giving them the same amount of cocaine. This is a collaboration with the Kennedy lab, Mabruk et al. What you see is the low responders get activated in red. The high responders are running all over the place. Very, very activated. If you do microdialysis to measure cocaine, dopamine in their brain, you see that the same dose of cocaine is triggering a lot more dopamine in their brain, and you can quantify it. More interesting is if you give them a chance to self-administer. This is Mike Emery, currently in the lab. You see, we've seen it repeatedly, but this is a great example where you see the first day the high responders take the max they're going to take. They love it. They stay up high the entire time of self-administration. The low responders start slow. They build up their willingness to take drug, and then they settle down at a lower level. Some actually refuse to take drugs altogether. And the same for heroin. So it's not just psychostimulants, true for heroin as well. And the high responders show very different relationship to the drugs than the low responders. So Shelley Flagel, when she was in my lab, did these studies on sign tracking and goal tracking and showed that the high responders are sign trackers. That means they fall in love with the signs that indicate that a drug is going to come. Whereas the low responders use the sign just as information, and they care only about the goal. And in this study, showed that we showed that dopamine release coincides with the sign with the high responders. So they see the sign that says a drug is coming, and they release dopamine at that point. Not true for the low responder goal trackers. So the whole dynamic of what they respond to is really different based on this breeding paradigm. So overall, the high responder animals are very different, as I showed you, not only in self-administration, but in the bred animals, conversion to addiction, relapse. A lot of that was in Flagel et al. In PNAS. And sensitization, impulsive action, and as I showed you, sign tracking, goal tracking. So what it says is that we need to probably be thinking about why people take drugs. Does it come from sensation seeking or from stress reasons, that maybe the drug can act as a homogenizer, like because it has such a big impact on the brain in the moment. But there might be these individual differences that trigger different behaviors, and reactivity to the drug might re-emerge after the drug is off board or after extinction, and leave behind different neural and behavioral profiles that might require different treatments. So that is a central premise for us, that it matters how you get to something, whether it is the depression or drug-taking behavior, temperament matter, and it affects the brain signature. So I hope that I've convinced you that at least we have a decent model of the human tendencies for internalizing, externalizing disorders, with a set of animals that have a whole profile of greater anxiety, stress reactivity, and another set of animals that are more impulsive, sensation-seeking, drug-seeking animals, but that you can actually also trigger drug-taking behavior in the anxious animals. So a big question, currently funded by NIDA, with this group of collaborators, Watson, Lee, and A. Palmer, is to try and get at the genetics underlying these two behaviorally distinct phenotypes. And to do that, you need to not only look at the main lines, but to also cross them to get a heterozygous, cross them again, to get an F2 generation to reproduce kind of a normal distribution, so you can begin to correlate genetics, gene expression, and behavior, because the behavior now will get redistributed. It will not just be extreme. And in this breeding, where you wind up with hundreds of animals at the end, we measure locomotion, which is our initial test. We measure anxiety with elevated plus maze. We measure sign tracking, goal tracking. We also have some developmental measures. And the F1 animals, the parents that are heterozygous, behave exactly kind of in the middle between the two in anxiety and so on, whereas the F2 animals are now distributed almost like a normal distribution. So we used a host of technologies, genetic and genomic technologies, to begin to come up with targets, both genes and functions, that might distinguish those brains, focusing primarily on the hippocampus because of its role in stress, and the nucleus accumbens because of its role in reward. Whole genome sequencing, long-read PacBio, bulk RNA-seq, ATAC-seq, so ATAC-seq to look at chromatin accessibility in the brains and related to gene expression. Single nuclei, multi-omics, also these data are not like fully digested, analyzed, digested. We use spatial transcriptomics to look at anatomy. We're using Visium, so it is not truly single cell, it's multicellular. So we follow it up with this tool that Vivek Kumar in the Watson lab has really honed nicely, where we could do this multiplex single cell, multi-transcript HCR fish, and you can wash it and do another batch and another batch, so you can look at subcellular level specificity of multiple genes expressed together. And big picture, this training created really massive differences in gene expression in both the hippocampus and the nucleus accumbens between the high and the low responders. I'm showing you a volcano plot that shows the differences here from this paper by Hebdebauer and Hagenauer. And it's contrasted, say, with comparing males versus females. And you see the number of genes that pass criteria are in a couple of thousand, whereas we only get like 30 or 35 genes that really pass these same criteria for differences between males and females. So it's a very substantial difference in gene expression patterns in these two brain regions. These animals all originated from Sprague Dawley and just bred on this one simple behavior. Now you can triangulate between the genetics, the genomics, the behavior, and begin to identify targets that meet a bunch of criteria. We have a whole bunch of targets on chromosome 1, but we have others, as you can see, on 7, on 20, and they're all very interested in some of them actually are concordant with what is seen in externalizing behavior, especially related to nicotine, actually, findings in humans. We see also differences in cell types and functional pathways that are favored more in the low versus high responders. For example, microglia activity is greatly enhanced in those anxious low responder animals. Or immune responses versus growth and proliferation is enhanced in the high responder animals. One major target that has popped up as being really different, and that's again Hebdebauer, Hagenauer, our recent publication, is mitochondrial function. So the low responders, even though they are mostly sitting around not doing very much, are very show a lot of mitochondrial activity that shows fatty acid oxidation, increased electron transport chain, reactive oxygen species, really stressed mitochondria compared to much better feedback and control in the high responder animals. And the combination of gene expression and ataxy chromatin shows that a pop hit is peroxisome, the whole peroxisomal system, and a particular gene called PEX11A seems to be a big central gene here, that 11 alpha. It regulates peroxisomal maintenance and proliferation. It's a gatekeeper for fatty acid oxidation. It's very nicely segregated genetically. It's allelic variation between the high and low responder animals, and shows differences in gene expression where it's elevated in the low responder animals. And chromatin, it's more available, it's more open. The chromatin is more open. You see this higher peak of near PEX11A, again in the high responders. And spatial transcriptomics, which I'm not showing you, shows that to be true beyond in other brain regions. And then when you do the whole population with the crosses, you see that there is a correlation between how much of that gene you express. The more you express of it, the less you locomote in a new environment. So it's clearly a target. We have not yet targeted with something like CRISPR. But it's the kind of now we're ready to start targeting some of these pathways. I like drugs that are already used in humans, and one of our approaches is to use drugs like pioglitazone, which is a PPAR gamma agonist, which can target a number of these functions and has been shown to be antidepressant, and see if it has differential role in the high versus low responder animals. And related to that, to the mitochondrial dysregulation, is microglia dysregulation, which we also see in these anxious animals. These complement-related genes that are expressed in microglia are also elevated on a number of measurements, like in the hippocampus, in the nucleus accumbens, by PCR, by single-cell transcriptomics. And Pam Maris in the lab did this very painstaking morphological study in the hippocampus about resting shape of the microglia in the anxious low responders versus high responders. And the number of microglia was not different, but the arborization of microglia was different. So they had a lot more arborization, which is usually compatible with the idea that they're, like what happens when you are immune challenged. She also gave a drug, minocycline, which is known to inhibit this phenomenon in microglia, and looked at it behaviorally. It gave it in the drinking water. The high responders did not respond to it, but the low responders on multiple tests of depression became less depressed. So it shows how you can go from these kinds of analyses and some neuroscience relating it to kind of morphology and so on, and then try to come up with targets that don't work for all creatures, but work for some creatures to change their phenotype. Obviously, you can drill down more, do CRISPR, do that sort of stuff, but basically it's a proof of concept that this is leading us to maybe useful information to look at these differences in temperament and how they can inform reactivity to stress or to addiction. So what I took away from all these studies is that these temperaments really kind of are not a single circuit or single neuron effect, but they kind of alter the fabric of the brain, the balance of cell type, the energy of the brain, the activity in the brain, inflammation in the brain, and that all of that is contributing to stress reactivity, to choices, and then consequence. And we're in the middle of analyzing the impact, the differential impact of cocaine, for example, on these animals, but this is work in progress. So I want to quickly, in this line of animal, talk about resilience. And you might think of resilience as not like intrinsic quality where you're not that sensitive to stress or you bounce back. But one other alternative way is to think about it more as a balance between positive and negative forces, and that has a biological underpinning. And the question is, can we begin to understand and characterize factors and features? And can we actually produce or see resilience in these animals that are genetically very anxious and prone to stress and anxiety? And I'm going to give you sort of two quick approaches. Again, a lot of this stuff has been published, so I'm just going to give you a flavor of it. We have two general approaches. One is an enriched environment, which we tried both in adulthood and in adolescence, where you're adding toys, in this case, to a big cage, and the animals are in there playing every day, or giving fibroblast growth factor and actually asking if the two are related. Why FGF2? FGF2 came from human postmortem studies that we had done in the Pritzker Consortium that showed us brain-wide changes in depression, but highlighted the role of the fibroblast growth factor in depression as one of the most altered families, with FGF9, which is not so well known, proving to be being elevated in depression. And in animal studies, we showed it to be a pro-depressant and pro-anxiety factor. And its good twin, FGF2, which sadly is low in human depression brain, but when you give it to animals, acts as an antidepressant and an anxiolytic. And so the question is, can we take that information back to our genetic model of susceptibility and resilience and see if we can deploy them here? And so this is a series of studies down here, Perez, Aaron Kozak, Turner, and you can start simply and say, are there basal differences in this FGF2, in the good molecule, between the high and low responder? And the answer is yes. Naturally, the high responder animals that are more immune to stress have more FGF2 in their dentate gyrus. If you knock down FGF2 specifically in the dentate gyrus, you don't change the red animals, the anxious animal, but you decrease the anxiety in the typically resilient animals. So you can drop the amount of time in the elevated plus maze from pretty high to pretty comparable to the more anxious animals by knocking down with this viral vector. If you now do a more environmental approach of living in an enriched environment, you create a whole bunch of changes in gene expression, in neurogenesis, you enhance neurogenesis only in the genetically anxious animals. And only in those genetically anxious animals do you improve anxiety behavior. You make them less anxious. And that is blockable by an antagonist for the FGF receptors. It's not selective for FGF2, but it's an antagonist that blocks it, suggesting that at least part of the effect of an enriched environment is mediated by FGFs. And this, I'm going to go fast here. I'm not going to do it justice, but basically you can give FGF2 in the first day of life. Courtney Turner did that and showed you have a lifelong impact on the animals, including making the anxious animals less anxious all the way into adulthood. So in other words, FGF2 is one example of a resilience factor that can counteract genetic susceptibility. Very strong genetic susceptibility can be counteracted. And I imagine there are a lot more molecular candidates. We have done other experiments where there are other targets that need to be validated. So before I move away from these studies, I just want to remind you that temperament is genetic, but that resilience can be induced in genetically highly vulnerable animals, and that therefore we need to think about both genetic and non-genetic types of resilience. So in the remaining time, I want to tell you about a human study that we're doing, and it involves college freshmen, and we're taking advantage of the fact that the freshman year is naturally stressful. And we're asking who's going to emerge as susceptible, as, happy or having suffered from some kind of disruption, depression or anxiety symptom during the course of the year. So the idea is to get as much baseline data as possible, psychological and biological, to follow them through the year and ask about the incidence of depression or anxiety and get data from wearables. So the design, and a big question for us is, can we predict? So the design involves recruiting these kids in the summer before the start of the freshman year, getting a ton of baseline endocrine, SCID, etc., 18 different psychological tests on them, then following them regularly on mood and anxiety measures and salivary cortisol, digital phenotyping throughout the year, all the way through to September of the following year. Some also Trier social stress testing as a social challenge. And during the COVID-19 period, which we captured in the study, we had to stop doing the Trier social stress test. So COVID was kind of a natural experiment that happened during the course of the study, where the kids went from, playing Michigan, being at Michigan football games, to packing up and going home. And so we have cohorts, and some of them are clumped multiple years together, but we have a yearly cohort every year from these kids. And we measure I'm focusing on PHQ-9 measures of depression. And you can see at baseline, when they're first coming through the door in September, you see how the baseline went up, so that by 2021 academic year, 27% of the kids were depressed, or met criteria for depression, as they walked through the door. It has improved since then very nicely. And then in the straight lines is who gets depressed sometime during the course of the year, meets criteria for depression. And you can see the freshman year actually triggers depression symptoms. It peaked at around 34-35% in 2021 and 21-22, those 2 first years of the pandemic. It has improved since then. Can we look at gender differences? So here are the females, pre-pandemic, the first 2 years of the pandemic, and since. You can see this rise, very sharp, 42% of the females, the first 2 years of depression, met criteria for depression that was not only a single time, it was chronic, it was lasting. We followed up in junior year, a lot of them were still depressed and anxious. And then they have improved, which is good news. Here's the males. They didn't seem nearly as affected during the first 2 years of depression. 3-to-1 difference in rates of depression at that moment. But now the males are not doing so well. So these are kids that were like in middle school during the pandemic, and those males are on a par with females in their rates of depression. So I think it's kind of worth noting what's going on with young males. During the pandemic year, like within that cohort, pre and post-pandemic, big rise, as you might expect, in anxiety, in depression, big decrease in number of steps per day. So you see this huge drop when they went home. But to me, what is remarkable is throughout the rest of the year, and even when they came back here as sophomores, so this is the beginning of freshman year, this is the same cohort, the beginning of the sophomore year, they're not moving. They really decreased their movement, and they were also as much more depressed. So is it genetic? So if you look at cohort before the pandemic, we calculated an MDD polygenic risk score, and actually it was kind of respectable, a few percent, predicts a few percent of the variance. But if you really had a low polygenic risk score, it didn't look like you were at high risk. But during the pandemic, that all disappeared. On every year of the pandemic, for the first about 3 years, absolutely no relationship between polygenic risk score and risk of depression. And there is like a host of people who had low genetic risk, but still showed depression, mostly females. This is mostly females who became depressed in spite of low genetic risk. But of course, there are people who have high genetic risk and are resilient. They never got depressed, whether it's pre-pandemic or during the pandemic. So obviously, this polygenic risk score is not capturing something important that the pandemic did to people. So is there anything else that can be predictive? So we had a host of psychological tests, and it's reported here in Turner, Khalil et al. Huzaifa Khalil used machine learning and took a whole host of the measures that we have. Some are state, some are trait, some are family history, and derived something we call the affect score. And the affect score turned out to be really highly predictive of the R is 0.67, so it predicts like half the variance. The odds ratios are higher than 10 in this cohort, showing that if you have a high affect score, as computed in like September at the start of the year, your odds of becoming depressed and anxious, which I'm not showing it to you, clinically significant depression and anxiety is likely. But since this was done on a cohort where it was validated, we have looked at it since then on several years of cohorts, 22 through 25, and you can see it's still very highly predictive tool. So again, we can take a young person at the start of their freshman year, and the odds ratio are like over 14. So it's a very strong tool for predicting future depression and future anxiety. So is there anything else that we can use? And I told you that we do the Trier social stress test. Probably you're familiar with it. You have to stand up in front of people, give a speech about your dream job, and then count backwards from 10 to 1,022 in steps of 13, which stresses people out. And I said something about stress fitness. So as a group, these young people, are they showing stress fitness? And the answer is yes. It's really lovely. There is a little increase in ACTH at the start when you put the IV, but it goes now nicely. They move to another room. They do their speech. They do the Trier social stress test. You see this nice sharp rise in ACTH, followed by court, and then beautiful shutdown. If you look at it by sex, you see the males have a somewhat higher response than females, but you see this odd group here in green, and that is women on birth control. In the blood, we measure these very high and flat level. We think maybe it's just interacting or doing something to binding protein, the birth control. But notably to me is that it's not just a measurement artifact, because ACTH is hugely depressed. So that's high level of court is really suppressing the ACTH response. So, something to think about, but going forward in these analyses, we did not include the females on birth control. So how does the TSST perform when people are currently anxious and depressed? This is current depression, and you can see here now red is control and blue is depressed. You can see both the court response, the ACTH response, and the court response are significantly lower in those kids who are currently depressed at the time of the Trier social stress test. So not mounting a good stress response. Can we use it to predict anything? And it's not easy to use it. It was done over a stretch of time. at the start of the year. But one of the things that seem to be predictive is of future anxiety. These are now kids who have never been anxious before. They were not anxious at the time of the Trier social stress test, but they will become anxious later in the course of the year. As a group, they had higher levels of cohort. If you separate them between males and females, this is 132 males, and you're looking at follow-up anxiety. There is no difference between the ones who will become anxious and those who will not. But in the females, there is really a very, very clear difference. In other words, even if you've never been anxious before and you're not currently anxious, if you have high response to cohort, this is a sign that you might become anxious during the course of the year. And this is how it translates if you combine the affect score that I told you is very predictive and this endocrine measure. You see that if you're low, if you're female and you're low on both measures, you have 5% chance of being anxious. If you're high on both, you have 52% chance of being anxious. And if you just have the affect score, sorry, that's my timer. I timed myself, so I'm late. It's somewhere in between. So, so we can predict to some extent who's fit and who's going to be vulnerable. And it makes you wonder if we can do something about it. And we're in very early stages, but we're interested in using cognitive behavioral therapy. This is a collaboration with Faith Gunning and Francis Lee at Weill Cornell, using an app they call the Maya app that they validated in a clinical population with both clinical measurements and neuroimaging. And we're starting to pilot it. We have encouraging early pilot data, but we have a big cohort, much bigger cohort, that we're testing now. So we stay tuned, but the idea would be if we can predict, can we prevent? That's really the hope. So the Michigan freshman study, and it's ongoing, told us that, we can capture the impact of stress in a naturalistic setting. We can see different types of stressor. The freshman stress versus the pandemic stress played out really differently. We saw male-female differences. We saw the role of genetics show up early, disappear. It's showing back up again. We can predict using this affect score and to some extent with some endocrine measures that we relate to kind of stress fitness. And this idea, again, that there is maybe more than one kind of resilience, that there is a natural kind of genetic resilience, but there is some other types of resilience that emerge in people who are genetically disadvantaged but still never budge in the face of stressors. So, I'm going to wrap up by saying that I've spent a lot of time now thinking about resilience and its genetic, but also the critical periods that matter and how we can combine non-genetic resilience to enhance or counter genetic susceptibility, that it continues throughout life, but there are always periods of possibility, especially early in life. But something I said earlier on, which is that specific experiences also matter, and that we need to be thinking about how they're constantly remodeling the brain, whether it's sensitization to drugs or repeated drug use or repeated stress, and how to interfere there so that you don't get on the wrong path. But I think what I hope that I have conveyed to you is that by kind of looking at individual differences and finding animal models and beginning to get at some of the underlying biology, that we can actively think more about prevention and intervention, and that we can adopt some of the approaches that have been used in other domains, like the Framingham study, where they defined both the individual risk factors but also the combination of risk factors that led to cardiovascular disease, and targeted them and actually succeeded very nicely for over the second part, at least, of the 20th century, to dropping it, in part through these prevention efforts. Some of them with drug treatments, like we take cholesterol drugs, we take depression drugs. Why don't we have drugs that are preventive in psychiatry, as well as other treatment? So my hope is that we think more actively about understanding stress and resilience and preventing not only the initial risk, like don't take drugs, which of course is important, but we're always going to get exposed to stress and trauma. But there are multiple occasions, primary, secondary, tertiary, of preventing long durations, preventing chronicity, reinterpreting, intervening at critical moment, that I think could be targeted. And if we could do a better job of figuring out who's at risk and how to engage these resilience mechanisms, I hope that we can do something about the mental health crisis that we are facing, especially in young people. So I want to leave you with this thought. It's a bowl that I have. It represents kintsugi. It's a Japanese art where it can take a cracked object and repair it in a way that doesn't necessarily try to hide the crack, but show how beautiful it can be after repair. And I think that we can all be cracked and beautiful at the same time. So thank you. I cannot see a single person or single thing. I'm blind. But okay.
So SSRIs are also anti-inflammatory from my understanding, serotonin. And you also find increased microglial activity.
Right.
Can you say more about how strong the role of inflammation may be in mood disorders?
I mean, I can speculate, but I showed you the data I have. But I firmly believe that what I learned broadly is to not think too precisely. It's just serotonin reuptake, right? But to think about the whole context, including inflammation in glia and so on. And that is a target that we need to measure and think about and see and match the treatment, like which SSRI is better for this kind of inflammatory phenotype. How do we capture that, like with proteomics, with exosomes in real humans, and how can we combine the information about the metabolic side, the inflammatory side, in our diagnosis, in our treatment selection? So are all the SSRIs equal? Are there some that are better? Do they work differently with different people? I hope that some of you who are much better at this than I am can build on that and do something with it,
Thank you.
Terrific talk, as always. But so it's the first thing, and I love the way that you're ending the presentation throughout the whole life. But it's just sort of like we started the ABCD study 10 years ago. So these kids are going to be going into college and are deeply, deeply phenotyped, including how was the reaction through the COVID pandemic.
Yes.
With an extremely important variable that per se you haven't spoken about, because in a way it's already too complex, which is your social support family, which can determine how you go. So I'm planting it in your brain because it would be perfect to actually test your hypothesis. But then let me ask you a question. And I am going to surprise you about it, because I completely agree with you that we need to transform the way that we look at prevention in psychiatric diseases in general, and from learning what we know about neuroscience. And we have tremendous neuroplasticity. And so there's an enormous amount of interest for different tools to address neuroplasticity. But one that has also polarized the whole field is that of psychedelic drugs. And there is preclinical data in animal models that early use of psychedelic drugs at very low content can actually enhance and counteract aberrant exposure. So what's your thinking, and very relevant to the question of serotonin, what's your thinking about psychedelic drugs? Have you thought about getting them involved in your paradigms?
Yeah. First, let me say something about your first part, which is that we think, and we're very interested in the the role of context and family support in resilience, especially in susceptible people. And we have some data, and I didn't have time to show it, but social support, self-reported perceived social support, is really an important component in protecting especially genetically susceptible people against, and it became more important during the pandemic. So you could see the difference in enlarging the role of social support becoming increasingly important during the pandemic. So absolutely, and I think understanding more about mechanisms there, and we even see it in our little creatures, where having buddies of their own kind creates a very different set of results than having, the opposing line in the neighborhood, in the same room even as we're doing the testing. It really matters who is around you in these temperament studies. With the psychedelics, I find it fascinating as a way to, I am very interested in critical periods, not only the way we understand them as childhood, adolescence, and so on, but plasticity critical periods before the system gels down too hard. Like in the social defeat paradigm, you don't get the emergence of susceptibility and resilience the first day. It takes 4 or 5 days of social defeat before the animals emerge as divergent. Meanwhile, the networks are biased to begin with in one way or another. The PVN is different and so on. The question is, can you intervene early on and rewrite the program? So if there are tools, and there are such tools of rewriting the program briefly so that it doesn't gel and reopen, then I think it's very cool. But my thought would be to be more precise, both about the people, but the timing. Not to just use it as a blanket. So if I had infinite time, life resources, I would try and work out what is the moment of an experience, or as it repeats, like as PTSD is becoming a part of you, what is the right moment to use psychedelics? Is it any time? Or is it the first time you have the crisis? That kind of stuff, I think, is needed. So it's a little more nuanced than let's just give psychedelics. But, I moderated a session with the Dalai Lama once at the Society for Neuroscience and asked him about how he believes in drugs to help people. And he said, anything to help with the troublemakers is okay. So anything to help with the troublemakers is worth trying. Yeah.
Hello. I'm from the Department of Perioperative Medicine, so we're really interested in pain. I was kind of curious if you saw any difference in pain if you assayed this or not, between the high and low responders, and also if you saw any difference in treatment resistance potentially in like giving the high and low responders like methadone or other opioid.
The only time, such a good question, and it's like embarrassing that I really don't have a good answer for it. The only time we tried, we had technical problems because they have other features that are different, like skin thickness, things like that make it harder because they've been bred for a while. So the short answer is I don't have a very good answer to you. The only time we did it was actually in the context of the chronic social defeat stress, which I did not really talk about. But this is done in inbred mice with the same genetic background, and they're just being exposed to an attack. And they emerge as susceptible or resilient. It's the Eric Nestler model. And the ones that emerge as susceptible show changes in glucocorticoids, but also changes in pain sensitivity. So pain sensitivity does come along with susceptibility and interacts with the stress system. And probably the opioids are very important in that. But I think it's definitely an interesting direction to go. Broadly, the question of temperament and pain sensitivity, I think, is a very interesting one.
Thank you.
Thank you, Huda, for just an amazing talk and visit. Huda will be around at the reception. Please join us for some light refreshments outside and more chance to talk to Huda. Thanks again.
Thank you.